Can everybody hear me? Yes? Yes? Good. All right. Great. Good morning. I'm really delighted to be here and thank the Planning Committee for gathering such an important forum on such a really critical topic. I'm going to be talking about understanding the dynamic relationship between genetics, environment, and early childhood development on the risk of obesity. What I'd like to do today is just explore some of this emerging science. I wish that I could tell you that it has been confirmed and we are ready to act on it. But instead, what I can do is show you some emerging properties of this evidence, and we can examine potential ideas for what might be modifiable or able to be mitigated as we examine innovative solutions for pediatric obesity prevention. So I'm going to move now. We're moving from a lot of numbers to diving deep into understanding this dynamic interaction between genetics, environment, and development. So consider this. Consider that we have about 22 to 24,000 active genes, but Pinot Noir, the wine that you might have had last night, has 29,000. So more doesn't necessarily mean more functional complexity. Our genetic code is a record of our past as well. We have more than 120,000 silent genes. And this is a record of our present, but what determines our future? And if 99.9% of all human beings have the same genetics, why do we see so many differences? Epigenetics is one way to look at this important question. Raise your hand if you've ever heard this term, epigenetics. Oh, this is good. This is about 50% of the audience. So the way that I'm defining epigenetics as we move forward in this part of the presentation is changes in gene expression via post-translational and post-transcriptional modifications. So think of it this way. You can't change your genes, but you can change the way your genes behave. So when you're thinking about this notion, for those of you who haven't been thinking about epigenetics, often we use the metaphor of music. So you think about the notes on the page as your genetics, and you think about the markings made by the composer or maybe the conductor as the epigenetics. So some markings are in ink, that's permanent, or what we call stable marks. And others are in pencil. In fact, they're so evanescent, you can see that I didn't even finish my statement there. So if it's in pencil, that means they're called unstable marks. These are the marks that we believe are modifiable. And in fact, even the stable marks, it isn't clear if we can intervene or not. So when we talk about the music that then is created after you create the notes, the genes, and the marks either in pen or pencil, which is that metaphor of the epigenetics above the notes, it creates the music. And that's what we mean when we talk about the phenotype. So if we apply this to living species, in all living species, we are adaptive. We are intended to be an adaptive species. We come out not fully formed, but we have received cues as we've been developing. And then we respond to the environment. So that means some have termed this the social life of genes. It could be short-acting or long-acting. So I'm going to present now the case of the killer bees. So what they've done, scientists have done, is they've taken the larva of the docile European bee, and they have put them into the hive of the killer bee. So the genes are exactly the same as the docile bee. But when the larva is placed inside of the killer bee hive, their behavior, their phenotype, looks identical to the killer bee. So the genes didn't change, but the expression of the genes did. So how do we apply that to people? Well, in February, the Institute of Medicine held a workshop. And what we did was we looked at the role of epigenetics and the opportunities for intervention. So I'd like to thank Ann Yachtine, who's in the audience, and she was the director of this workshop, and also Leanne Birch, who was part of the planning committee. So here is what we did. We looked at this idea of where does plasticity and reversibility, where is the greatest opportunity for that? And so here along the x-axis, we're looking at time. We start from preconception gestation, infancy, early childhood, adolescence, and adulthood, recognizing that these early exposures often are manifest much later in time. The opportunity for plasticity and reversibility appears to be greatest in these earlier years, but you still have opportunities as you age. And then you have to consider that exposures happen at multi-levels in the environment. So you look at metabolism and physiology. You look at that at the level also of the child. You look at the exposures and the level at the family level, community, and environment. The workshop wasn't intended to be all-inclusive. We only had a day and a half. But what we did was we focused on very specific areas. So we looked at this epigenetics. We were considering appetite and satiety, and specifically rapid infant growth, maternal nutrition and physical activity, both maternal and paternal nutrition and other factors such as stress, early in this preconception and gestational period. And then your exposure to the environment at a much larger level, so health and economic disparities. I'm not reviewing the entire workshop today. Instead, I'll focus on maternal nutrition and paternal nutrition. So we start with maternal nutrition. So raise your hand if you know about the agouti mice, the agouti gene. And that's about 30% of you in the group. So what's really fascinating about these mice is that they are genetically identical. The only thing that's different about them is that they were exposed to a different diet when in utero. So the yellow mouse that has the obese phenotype just received a regular diet. But this more lean phenotypic brown mice, the diet of that mother during gestation was supplemented with choline, folic acid, and vitamin B12. This affected the methylation at the agouti locus. So just a word on epigenetics. There are many ways to consider what epigenetics is. I'm going to be focusing specifically on methylation as a mechanism for epigenetics. And as we talk about methylation, it's confusing. And the reason why this is confusing is it depends on which loci is methylated as to the later downstream effect about how that affects expression. So when you think about epigenetics, you need to know where the methylation occurred, either at the gene promoter region or in the gene body, and also which tissue was it expressed in. I'm not going to dive deep into all those scientific differentiations. I just want you to be aware of that as you go forward and learn more about epigenetics. So here we're just looking at epigenetics with methylation marks. And I think what's really interesting here is that these agouti mice, these typically, just with regular exposure, they will have an obese phenotype. But it can be mitigated, it can be modified by changing that maternal diet. So if you change the maternal diet and have that dietary supplement during pregnancy and also lactation in nursing, then you can see that the majority of these pups have the phenotype of the lean mouse. Whereas if you didn't have dietary supplementation, it's inverse. And the majority of these mice have the obese phenotype. Clearly, maternal nutrition is really important. So how does this apply to other models and also humans? So multiple models have been assessed. It's difficult to control nutrition for human beings, to actually measure and monitor what we eat well. For that reason, many models with very controlled environmental settings have been tested. So what I've done here is I've given you an overview. This relates to many different animal models, including humans, and high-fat diets during pregnancy. So the maternal environment here is a high-fat diet. So a word about that, we know fat is not bad. It is too much fat, or the type of fat that you're exposed to here. And we also know that many of these studies looked at what we call the Western diet. So not only high in fat, but also in sugar and salt. So if you think about this maternal environment, many studies have demonstrated a differential gene expression due to these epigenetic changes measured by methylation in these specific genes. What's important about these genes is that they are responsible for lipid metabolism, glucose metabolism, and appetite, either stimulation or suppression. These are associations, these studies, showing that a high-fat diet during pregnancy is associated with methylation changes and differences in these genes with this biologic plausibility for then leading to a phenotype of obesity, and then associated with that infant phenotype of regulation of food, carbohydrate, and lipid metabolism that can be dysregulated. But it's not only the human DNA that seem to utilize and be associated with these epigenetic changes. It's also our microbiome. So raise your hand if you know that word. You're familiar with that? And that's 90% of you here. So I love this fact around our kitchen table that the majority of our DNA is non-human in our body. We have tenfold more DNA that is non-human in our body, and we've got this unique symbiotic relationship with all of these microbes. So we can look at epigenetic changes in those microbes as well. And here are some of the things that have been discovered. So maternal obesity in breastfeeding mothers affects the infant microbiome, that affects adiposity, and it's associated with these epigenetic changes, these methylation modifications. A mother on a Western diet more often has a shift in that microbiome species that affects metabolism and the inflammatory response. And composition of the microbiome also affects leptin and insulin levels, as well as the N6 to N3 fatty acid ratios. So exposure to a maternal Western diet through breast milk appears to pattern the microbiome and affect a pro-inflammatory state in infants. Something that's so critical about children is you might have experienced this exposure early in your development, but the manifestation of the phenotype occurs later. And that's why these longitudinal trials need to look over time to try to better understand what we see now is correlation. Much of the studies that are underway are now looking at causation. And it's not only the mother, the father matters too. So when you look at paternal nutrition, this science is interesting and I think less discussed. So there appears to be an epigenetic mediation through sperm and the paternal line. So a high-fat paternal diet increases beta cell dysfunction in female offspring. And it appears that paternal obesity affects the metabolic and reproductive health of the offspring for multiple generations. So when you look at this type of work that has been done, you can see that here's the maternal obesity affecting cellular functioning and specifically mitochondria. And here is the paternal contribution with paternal obesity with a metabolic syndrome affecting DNA or chromatin damage, perturbing these epigenetic reprogramming of the embryo. And it's been shown in mice, less clearly in humans, that this seems to affect multiple generations. So what can we learn from epigenetics to better understand childhood obesity? Godfrey examined longitudinal cohorts of children and epigenetic signatures of their umbilical cord tissue, and then looked later at fat distribution using a DEXA scan at children that were age 9, and noted these findings, that methylation patterns were different at these specific genes. And specifically, the RXRA was confirmed as differentially methylated for those children with later increased adiposity in an independent sample of children. There was no causal mechanism identified, just correlation. But it appeared that these changed methylation patterns was associated with later childhood adiposity. And the authors concluded that perinatal epigenetics could potentially be an opportunity to assess individual vulnerability to childhood obesity. What else could we learn as we apply this emerging science of epigenetics? So I present some of the work that we're doing right now in a larger trial. We've got a 7-year randomized controlled trial to look at family-based, community-centered interventions to see if we can prevent obesity in preschool-aged children. And as a proof of principle, we looked at saliva from our formative phase and also some of our baseline participants. And we looked at saliva because we work with preschool-aged children, most of whom are from underserved communities. We collect our data in the community setting as well. So it's an easily accessible tissue, and we wanted to know what could we learn from this in terms of epigenetic differentiations. Were there different signatures? And I'll explain how we think this could be useful. So we only have enrolled children to participate in this study if they are not yet obese, but are at high risk for obesity. So that means their BMI is greater than the 50th percentile, but less than the 95th, that at least one parent is obese. They are all on some sort of supplemented state assistance program, such as WIC or SNAP. And we wanted to know if we look at that maternal BMI, for children who are not yet obese but are at risk for later obesity, could we see differential salivary methylation patterns? And we focused on mostly our Latino families. About 90% of our sample are Latino. So here is what we've found in terms of differential patterns in epigenetics. We see that they are different, predicted by the BMI of the mother. So these children, again, not yet obese. And it seems to point to some of these types of mechanisms. Most of these are going to make a lot of sense because they've got a biologic plausibility. So, for example, we see differential methylation in this melanin concentrating hormone receptor. This is involved in regulation of food consumption. We see differences in this gene, which allows you to regulate the amount of cholesterol, as well as the CCL5 gene, which is involved in the inflammation response. And interestingly, this complex and this gene, which is involved in the inflammation complex that's involved in gut microbiome sensing capabilities. So why do I bring this up? I think we believe that we can use epigenetics to help guide, number one, where we potentially might focus to intervene, what is modifiable, and number two, how we can better understand who's at risk and also potential mechanisms. So to summarize, maternal malnourishment, either over or under, can increase the risk for childhood obesity. Animal models have examined maternal supplements such as choline that change methylation patterns and is able to reverse early metabolic dysfunction. It's important to note that these relationships depend on age, gender, and maternal nutritional status. Second, regardless of maternal BMI, a high-fat maternal diet during pregnancy is associated with an increased inflammatory cytokine production in the fetus and also in the placenta. And some of these effects appear to be mitigated if postnatal nutrition has normal fat composition. Another opportunity for intervention and prevention is paternal nutrition. If we see that it can increase the risk for childhood obesity, we could address this through improved paternal nutrition. So epigenetics could provide an approach to identify at-risk children, as well as understand mechanisms to shed light on factors that could be potentially mitigated. I'd like to thank our IOM workshop planning committee. You can see all of the individuals that participated. I've just given you a small taste, but the book associated with the workshop was just published at the end of September, and it has many more details. Thank you very much.